Hello, everyone, and welcome to today's webinar, Advancing Astrocyte Research with Novel Tools for Characterization, Isolation, and Cultivation. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more about our sponsor, please visit www.miltenyibiotec.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. I encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Melanie Jungblut, the Senior Project Manager and Group Leader of Neuroscience at Miltenyi Biotec. For the complete biography on our speaker, please visit the Biography tab at the top of your screen. Melanie will now begin her presentation.
So welcome everybody to our webinar about advancing astrocyte research with novel tools for characterization, isolation, and cultivation. We will talk today about the anti-GLAST and anti-AXA2 antibodies. So these are the first antibodies which allow for a specific detection of living astrocytes. We will first show you how the antibodies were generated and then go into the details for the characterization of both antibodies. Afterwards, we will introduce different workflows, including tissue dissociation and separation of neonatal and adult astrocytes, and then talk about some downstream applications of isolated astrocytes. In addition, we will introduce you into the analysis and separation of adult neural stem cells, which is a rare type of cells that shows astrocyte characteristics. So, the anti-GLAST antibody was the first antibody that allows for a specific detection and separation of living astrocytes, as it detects an extracellular epitope of the glutamate transporter GLAST, which is also called EA81. We call this antibody also AXA1, which means astrocyte cell surface antigen 1. The second antibody that was generated was the anti-AXA2 antibody. This antibody was generated in rat and has the isotype rat IgG2b. When the antibody was generated, the official name of the target was not known, and therefore it was called anti-AXA2. But in the meanwhile, we know now that the antibody detects the ATP1B2, which is the sodium-potassium transporting ATPase subunit beta-2. Both antibodies were generated by immunization. For the GLAST-specific antibody, transfectants expressing GLAST were generated and served as immunogen. The GLAST knockout mice were immunized several times with these transfectants, and then the lymph nodes were isolated from the animals and fused with myeloma cells. The resulting myeloma cell clones were then screened for their specific GLAST antigen recognition, and then the positive clones were expanded and subcloned. Also, the anti-AXA2 antibody was generated by immunization, and in this case, GFAP positive cells that we isolated from GFAP eGFP mice were used for immunization, and rats were immunized. So now we come to the characterization of both antibodies. In this slide, you see an immunohistochemical staining of a sagittal brain section of P1 wild-type mice with the anti-GLAST antibody in A and B. So in that case, GLAST immunoreactivity was detected throughout the whole brain. But in contrast, no staining was observed in brain sections derived from GLAST knockout mice, as you see in C. So it was also investigated whether the antibody can be used for labeling of living astrocytes by flow cytometry, that you see in D and E. And you see in D that a clear GLAST-positive population was detected in case of the wild-type mice, whereas no signal was obtained in case of the GLAST knockout mice. And we observed also the same by Western blot analysis, as you see in F. Two bands corresponding to the GLAST monomer and to the dimer were detected in wild-type mice, whereas no corresponding signal was seen in the knockout animals. So, and that means that these results demonstrate that anti-AXA2 antibody specifically detects an extracellular domain of the native as well as denaturated GLAST protein. So on this slide, the anti-AXA1 staining is shown in 1 or 2-week-old primary mixed neural cell cultures. And here the co-staining for GLAST and the astrocyte-specific antibody GFAP in A and B show that cells marked by these antibodies are all detected by the anti-GLAST antibody in 7 as well as 14-day-old cultures. And you can see that the anti-GLAST antibody stains the surface of the cells and therefore nicely surrounds the intracellular GFAP staining. And therefore it stains the typical branched astrocyte morphology that the GFAP staining does not show. And in 14-day-old cell cultures, we detected also astrocytes that were GLAST positive but GFAP negative, as you can see. And this is because these astrocytes are no longer reactive and therefore they do not show GFAP expression, but they still show GLAST expression. And that's what we saw, that the GLAST is a broader marker for astrocytes than GFAP. And we performed also co-stainings with additional astrocyte markers such as BLBP, RC2, GS, and nestin. And all BLBP, RC2, and GS, and nestin positive cells with astrocyte morphology were also detected by the anti-AXA2 antibody. And that means that the anti-AXA1 antibody shows specific detection of all cells detected by different astrocyte-specific markers. So, the expression of GLAST was also investigated on other cell types as oligodendrocytes and neurons, and we found no co-localization with the oligodendrocyte-specific marker MOC and O4, as you see here on these pictures. Furthermore, we could also not detect any double positive immunoreactivity for GLAST and markers for PSA-NCAM positive neuronal progenitor cells, or NUN or MAP2. So taken together, in cultured neural cells, GLAST expression, as revealed by this anti-AXA1 antibody, was detected in virtually all cells, showing the typical astrocyte morphology and expression as the common astrocyte markers. And GLAST showed a broader expression than classic astrocyte markers like GFAP, and it was not found on other major neural cell types like oligodendrocytes and neurons. So the next slide shows the immunohistochemical staining of GLAST expression in different mouse brain areas. And you see that GLAST-positive cells could be detected in all brain areas with highest expression levels in the cerebellum, as shown in A to F. And here the strongest GLAST expression was visible in the molecular layer of the cerebellum and in the Purkinje cell layer that you see, for example, in the image D. So, and this is because of the GLAST-positive Bergmann glia, which are located in the Purkinje cell layer and extend their processes throughout the molecular layer. And in comparison to the molecular layer, the cranial cell layer showed a much lower expression of GLAST, and the white matter showed almost no GLAST expression. And in the cerebral cortex, that's shown in G2I, we detected a punctate GLAST expression because of the cell surface labeling here that you see with GLAST. And in the corpus callosum, GLAST was neither expressed by oligodendrocyte progenitors expressing NG2, as shown in J2L, nor was it expressed by mature oligodendrocytes that were identified by the expression of carbonic anhydrase 2, that's shown in M2O. We then investigated the GLAST expression also in the two neurogenic brain regions of the brain, the subventricular zone and the dentin gyrus of the hippocampus. So in these regions, you find stem cells that show astrocyte characteristics, and it's also described that these cells express astrocyte markers. And here in A to C, you see that GLAST and GFAP are widely expressed and partly overlapped in the periventricular zone. So and here in this periventricular zone, neural stem cells were labeled by TD tomato by postnatal in vivo electroporation. And it can be seen that the electroporated TD tomato positive cells express both GLAST and GFAP, so that you see in A to C. And that means that the anti-AXA1 antibody identifies the neural stem cells in the periventricular region of the postnatal forebrain. And in D, we see that GLAST is expressed on astroglial cells that form scaffolds along the rostral migratory stream, but there's no expression detected in the PSA-NCAM positive neuroblasts. And in the second neurogenic region, in the dentate gyrus of the hippocampus, GLAST was strongly expressed throughout the granule cell layer, so that you see in G2I. And it was associated with the GFAP positive processes. And newly generated neurons that were identified by the expression of PSA-NCAM were also GLAST positive. But this is in agreement with the glial nature of the hippocampal neuronal progenitors. So in conclusion, we can here say that the anti-GLAST AXA1 antibody detects the neural stem cells in the periventricular zone, and it also detects the tunnel astrocytes in the rostral migratory stream, but the PSA-NCAM positive neuroblasts were not detected. In the next slide, we see the cross-reactivity of the antibody to rat and human GLAST that was analyzed by immunohistochemistry, flow cytometry, and Western blot. And you can see in figure A, the antibody detects the astrocytes in both human and rat brain sections, and it also shows the same staining pattern in the cerebellum as observed in the mouse brain section. So bright staining in the molecular stain layer and the dimmer staining in the granule cell layer. And also the Western blot analysis of total rat and human brain protein revealed different bands, and these bands correspond to the monomer and to the dimer, as you see in B. And then in C, there's also a flow cytometric analysis shown of dissociated P7 rat brain, and here we detected 19.5% of GLAST positive cells. So as a summary for the anti-GLAST AXA1 antibody, we can say that this antibody was the first monoclonal antibody that allows a specific detection of living astrocytes with the isotype mouse IgG2A. This antibody detects an extracellular epitope of the glutamate transporter GLAST, also called EA81. We showed you that it labels all GFAP, GS, S100 beta, BLBP positive astrocytes. It also labels reactive astrocytes, radial glia, the neural stem cells, also the Müller glia in the retina and the Bergman glia. So it's a very broad astrocyte marker. It's also expressed by quiescent, so GFAP negative astrocytes, and it's cross-reactive for mouse, rat, and human. So next we show you the detailed characterization of the second astrocyte-specific antibody, the anti-AXA2 antibody. So the first characterization is shown by immunocytochemistry, and here again mixed neonatal neural cell cultures were used and stained with different neural cell-specific antibodies. So you see here the anti-AXA2 staining, which nicely shows the staining of the cells that show typical astrocyte morphology. And the co-staining with anti-GLAST shows that both markers are co-expressed here on the majority of cells. But it's also obvious that there are different expression levels of both markers. So furthermore, we could show that the AXA2 expression is not detected on CD11B positive microglia, so no overlap with this marker. There was no overlap with O4 staining, so no expression of AXA2 by O4 positive oligodendrocytes. And we also did not detect AXA2 expression on PSA-NCAM positive neuronal precursors. So on the next slide, we see the immunohistochemical analysis of AXA2 expression in the cerebellum. And you see here in the first row that AXA2 detects GFAP positive astrocytes, and in the second row that it also detects the S100 beta positive astrocytes, but it shows lower expression in the Bergmann glia in comparison to S100 beta. And in the third row, you see that AXA2 overlaps with GLAST in the cerebellum, but it shows a higher expression of the granule cell layer in comparison to GLAST. So there are different expression levels of all these markers. We also investigated AXA2 expression in the striatum, and here the anti-AXA2 antibody shows a clear co-expression with GLAST, as you see in the upper row, and no co-localization with the oligodendrocyte marker MBP in the second row, and it also showed no co-expression with beta-3 tubulin, as you see in the third row, so beta-3 tubulin as a marker for neurons. So as a summary, we can say that the anti-AXA2 antibody specifically detects GFAP, S100 beta, and GLAST-positive astrocytes, but we did not detect any double positive immunoreactivity with oligodendrocytes or neurons. So the next slide shows the flow cytometric analysis of AXA2 expression in the neonatal mouse brain. And in A and B, it's shown that 40 to 50% of A2B5 positive cells show AXA2 expression, which shows that glial-committed precursor cells express the AXA2. But on the other hand, O4 positive oligodendrocytes, CD31 positive endothelial cells, and CD45, CD11b positive microglia show no expression of AXA2. So in G, we see that the majority of prominent positive cells, so more than 80%, show AXA2 expression in the neonatal mouse brain, as these are very early cells, so progenitors. And we see also co-staining of AXA2 and the ependymal cell marker CD24 on a fraction of cells. And in I and J, it's shown that PSA-NCAM positive neuronal precursors and GABAergic neurons that were analyzed in GAD67 EGFP transgenic mice did not show a clear co-expression of AXA2. And then in the adult mouse brain, we see again that A2B5 positive glial restricted precursors are partly AXA2 positive, and O4 positive oligodendrocytes, CD31 positive endothelial cells, and CD11b, CD45 positive microglia, and also PSA-NCAM positive neurons are devoid of any AXA2 expression. So, and in the adult mouse brain, the expression of AXA2 on prominent positive cells is not detected anymore, as you see in Q. So as a summary for the anti-AXA2 antibody, we can say that this was our second monoclonal antibody that allows detection of living astrocytes. So this antibody detects an extracellular epitope of ATP1B2, and it is co-expressed on GFAP, S100 beta, ALDH1L1, that was also analyzed, and GLAST-positive cells. So it's also widely expressed on astrocytes. And we could also show that this mark is expressed on quiescent GFAP-negative astrocytes and reactive astrocytes. So the anti-AXA2 antibody can be used as an antibody to label, in principle, all astrocytes in the brain. Okay. In the next part, we will show you now how to dissociate tissue and give you more information on the separation and analysis of astrocytes. Before cells can be separated, the tissue must be dissociated, and the prerequisite for proper cell separation is really the sample preparation and the generation of a single cell suspension with a high percentage of viable cells and a minimum of debris. So, and therefore, successful experiment really starts with a very well-prepared tissue dissociation. And for proper tissue dissociation, a combination of a mechanical disruption and an enzymatic digestion is needed. And the mechanical disruption in our case is performed with a gentle MACS dissociator and the unique dissociation tubes, which are operated by the gentle MACS with specific programs. And for the enzymatic part, we developed tissue-specific dissociation kits. And the combination of the gentle MACS with these tubes and the specialized dissociation kits then guarantees for the most gentle and efficient tissue dissociation and a high amount of viable cells and a minimum of debris after the tissue dissociation. For neurotissue dissociation, we developed kits optimized for its special needs of the neural tissue. We have here titrated enzymes, consistent activity from lot to lot, and these kits really give you reproducible results. We also give here detailed protocols, and it's also very important that these kits ensure the conserved epitopes for downstream applications, which is very important that your separation or analysis works afterwards. We have here different kits with different enzymes, and they are optimized for neonatal as well as adult tissues. For neonatal tissue, we have the neural tissue dissociation kit PNT, and there's also one kit that's optimized for postnatal neurons. And for the special needs of adult neural tissue, we developed the adult brain dissociation kit. How the workflow for tissue dissociation, separation, and further downstream application looks in detail depends on the age of mice and the cell type of interest you want to separate or analyze. So in case of animals up to P7, we have, as mentioned, the neural tissue dissociation kits PET or the neural tissue dissociation kit postnatal neurons. And when you work with animals older than P7, we recommend to use the adult brain dissociation kit. And this kit already includes the debris removal and an erythrocyte lysis that is needed in case of older tissue derived from older mice. So I just want to tell you a little bit more about the adult brain dissociation kit. This kit combines an optimized enzymatic and mechanical treatment. We use the gentle MACS octodissociator with heater for the dissociation, and the kit then includes also an efficient sample clearing afterwards. The workflow looks like this. You first cut the brain into sagittal slices, and then the optimized enzymatic and mechanical dissociation is performed using the gentle MACS octo with heater, which takes around 30 minutes. After that, you perform a cell debris removal and erythrocyte removal, and then you go to the cell separation. This takes altogether 1.5 hours. So here you see an example of a dissociated brain after the dissociation. So in the upper row, you see it directly after the dissociation, and although the dissociation was optimized as far as possible, we still see a consistent amount of debris, as you see in the first plot in the upper row in the forward side scatter. And also a high percentage of erythrocytes. And it's very important to deplete this debris and erythrocytes to perform a successful cell separation afterwards. And you see in the lower row and that after the cell debris and erythrocyte removal, the debris was efficiently depleted and also the erythrocytes, so that you have a high percentage of target cells. And then you can go on with your cell separation, which will be then really successful. So we now have a look on the separation and the analysis of the astrocytes. In case of the neonatal neural tissue, astrocytes can be either separated using the Anti-GLAST or the Anti-AXA2 microbead kit. And in case of adult brain, the Anti-AXA2 microbead kit is recommended. That's because the GLAST antigen is papain sensitive, and therefore the anti-GLAST microbead kit is not compatible with the adult brain dissociation kit. The principle of the separation is a positive selection. That means you incubate your microbeads with a cell suspension, and then the astrocyte-specific microbeads bind to your target cells. And when you then give the cell suspension onto a column, which is placed in a magnet, all the unlabeled cells run through, and the labeled cells stay inside the column. And then you remove the column from the magnet, you elute your cells, and then you have your positively enriched astrocytes. The workflow for the anti-GLAST AXA1 microbead kit looks like this. So you have to dissociate your brain with the anti-DKT. After that, labeling with the FCR blocking reagent is performed to prevent any unspecific blocking to the Fc receptor. Afterwards, you label with the anti-GLAST biotin conjugate. Then you perform a washing step, and then the labeling with the biotin microbeads is performed. So this is an indirect labeling. And after another washing step, you separate the cells with MS column, and you find your positively enriched astrocytes in the positive fraction. So this process takes about an hour. Here you see an example for such a separation. Before the separation, we detected here a percentage of around 17% of GLAST-positive astrocytes, which were enriched to a high percentage of 95% after the separation. And then these isolated astrocytes can also be cultivated, which is shown here after 3 days in vitro. A staining with the GFAP and GLAST antibody was performed, and you see that you have here a nice dense layer of pure astrocytes. The workflow of the anti-AXA2 microbead kit is a little bit different. Here the dissociation can be performed with the anti-DKP in case of neonatal mice, or ABDK in case of mice older than P7. Then we have again labeling with the FCR blocking reagent, and then it's a direct labeling with the anti-AXA2 microbeads. And after washing, you can go to the separation with MS columns. And as this is a direct labeling, it's a little bit faster and takes around 35 minutes. In this slide, you see here an example for a separation of neonatal astrocytes and adult astrocytes. In the upper row, the separation of neonatal astrocytes. Before the separation, we found here around 22% of astrocytes, a nice enrichment to around 97% after the separation. And in the lower row, an example for a separation of adult astrocytes, which were enriched from 30 to 90%. And you can cultivate neonatal as well as adult astrocytes. That's shown in the picture C and F here, and you get nice cultures. And we stained here for GFAP and GLAST, and you see that we have here a nice layer of GFAP GLAST double positive cells. This is another example of a cultivation of adult astrocytes after the isolation. Here 100,000 cells were cultivated in a 24-well in AstroMACS Medium on PLL laminin coating. And you see here really a very dense, nice layer of GFAP GLAST double positive cells. And in the lower row, you see that there was no contamination of MAP2 positive neurons. Only one MVP positive oligodendrocyte was detected here, and no contamination with CD11B positive microglia. We can also separate astrocytes by magnetic cell separation from distinct brain regions of adult brain. So here an example for a separation from cerebellum, cortex, and hippocampus is shown. And you see that in all cases, the astrocytes were enriched to a high percentage here of 95 to 97%. So this also works nicely. And the cells can also be cultivated. Here they were plated again in Max Planck Institute medium on PLL laminin coating. And in the upper row, you see the cultivated astrocytes that were isolated from the hippocampus and co-stained for GLAST and the AXA2. And here you see a nice expression of both markers. And again, you see that the cells have different expression levels for those markers. And in the lower row, you see staining for GFAP and GLAST. And here it becomes again obvious that the GLAST nicely detects all the astrocytes. Here you nicely see the astrocyte morphology, whereas the GFAP does not stain all the astrocytes. Okay, we can conclude now that for the magnetic cell separation, we have two options. The one option is the anti-GLAST AXA1 microbit kit, and this allows for separation of astrocytes from neonatal brain samples. As the GLAST epitope is papain sensitive, we have to use the neural tissue dissociation kit T in that case. And this is an indirect labeling via anti-GLAST biotin, antibiotin microbeads, and the antibody is cross-reactive for human, mouse, and rat. The second option is the anti-AXA2 microbead kit mouse. This is papain as well as trypsin resistant, so we can use all the different dissociation kits and also the adult brain dissociation kit. Therefore, this kit allows separation of astrocytes from neonatal and adult mouse brain. These are direct microbeads, but they can only be used to separate mouse astrocytes. So in the next part, I will introduce you into the analysis and separation of adult neural stem cells. Adult neural stem cells are a special type of cells, and they can be found in the two neurogenic brain regions of the adult brain. They are located once in the subventricular zone of the lateral ventricle, and they are also located in the dentate gyrus of the hippocampus. And today we will talk here about the stem cells of the subventricular zone. As mentioned before, these stem cells show astrocyte characteristics, and they also share astrocyte-specific markers like GLAST, and therefore they are also called stem cell astrocytes. Here we see the lineage, so the quiescent stem cell astrocytes can turn into activated astrocytes, stem cell astrocytes, which turn then into transit amplifying cells. And those transit amplifying cells turn into neuroblasts, which travel along the rostral migratory stream to the olfactory bulb, where they become mature olfactory bulb neurons. So the neural stem cells cannot be identified by one marker, and we need a combination of markers for their identification. And therefore, we developed a kit with a unique marker combination of the following markers for their identification. This kit includes the anti-GLAST antibody and furthermore the anti-plexin B2 as positive markers. And then we have CD24, CD45, and TER119 as negative markers. The workflow looks like this. So we first dissect the SVZ by whole mount preparation from adult mouse brain. Here we recommend to use the SVZ from 5 mice, so in total 10 SVZ. Then the tissue dissociation is performed with the neural tissue dissociation kit T in combination with the GentleMACS octodissociator with heaters. Afterwards, we have a debris removal step, and then we stain with the neural stem cell antibody cocktail. The sorting of the NSCS is then carried out with the MACS-Quant Tyto. This is a novel multiparametric cell sorting device that uses a microchip-based technology for sterile and gentle cell isolation. So as we need here a combination of markers, sorting with magnetic cell separation is not possible. And after sorting, cells are reanalyzed by flow cytometry, and they can be cultivated in a neurosphere assay. So this slide shows the gating strategy. As you see in A, the debris is first excluded in the forward side scatter, and then in B, the doublets are excluded, and afterwards also the dead cells, which are identified by PI staining. Afterwards, in D, the negative markers, the CD24, the CD45, and the TER119 are excluded. And then the gate is set onto the plexin positive cells, and then in the last step onto the GLAST positive neural stem cells. This slide now shows an example of a MACS-Quant Tyto-based separation. And in the first row, you see the non-sorted cells. In the second row, the positive target cell fraction. And in the third row, the negative cell fraction. And when we have a look at the sorted neural stem cells in the second row, we see that the CD24, CD45, and TER119 positive cells are efficiently depleted, and the plexin and GLAST positive cells are enriched to 97% and 98% respectively. So that means we have here really high purity of the neural stem cells in the positive fraction. So on this slide, we see the summary of the different parameters for the procedure. We have a total processing time of 3 hours when we prepare the SVZ of 5 mice. The sort duration is 30 minutes. We have the total cell number after dissociation is around 2 million. We can sort around 36,000 cells from the SVZ of 5 mice with a purity of over 95% and a viability of 93%. The yield of the cells is around 40%. So in the last part, we have now a look on the cultivation of the astrocytes as well as on the cultivation of the neural stem cells. We have the separated cells, and that can be now processed by different downstream applications like flow analysis, sequencing, or cultivation, for example. And in case of the cultivation, usually serum-containing homebrew media are used, and this leads to inconsistent results. And one drawback is also that the serum really affects the gene expression. So therefore, we developed the AstroMACS medium, and this is the only serum-free astrocyte medium on the market, and it can be used for neonatal as well as adult mouse and rat astrocytes. So this is a medium that contains the MAX Neuro medium, the MAX Neuro Pro 21, and in addition, a MAX AstroMACS supplement, which really improves the astrocyte culture. That we can also see on this slide. In case of cultivation of the cells in AstroMACS medium, as you see in the graph, you have a higher viable cell count after 7 days in all the different density in comparison to a cultivation in a MAX Neuro medium without the MAX AstroMACS supplement. And especially in case of the lower densities, the AstroMACS medium allows cultivation of astrocytes, whereas the cultivation in the other medium is in principle not possible anymore, as you see here in case of 10,000 cells. And also in case of the pictures, you see without the AstroMACS medium, there are only a few cells, whereas you get a very nice cell culture when you add the AstroMACS supplement. Our last slides are now about the cultivation of the isolated neural stem cells. And the isolated neural stem cells are here subjected to a neurosphere assay, and such a neurosphere assay examines the 3 characteristics of neural stem cells, and that means the self-renewal, the multipotency, and the proliferation. And the isolated cells were cultivated on ultra-low attachment plates in MAX Neuro medium, and this Neuro medium was supplemented with MAX Neuro Pro 21, EGF, and FGF2. And we see here in these pictures that the cultivated neural stem cells form the large number of neural stem cells and neurospheres under these conditions. And for the differentiation, we then plated the neurospheres on polyalysine coated 24 glass bottom plates without the growth factors. So after 7 days of cultivation, we fixed the cells and subjected them to immunocytochemical analysis using the astrocyte-specific antibody GFAP and nestin in that case. And we see here that the neural stem cells nicely differentiated into astrocytes and forms a lot of astrocytes here. And that's also shown in this picture, so staining of GFAP and nestin as astrocyte markers. And we see that we have a lot of astrocytes here. And furthermore, we also stain for a neuronal marker, MAP2, and for the oligodendrocyte marker, O4. And we could also show that the cells differentiated here into neurons as well as oligodendrocytes. So in summary, we can say that our neurosphere assay and the differentiation of the neurospheres show the capacity of the isolated cells for self-renewal and also shows their ability to differentiate. So I now come to the summary of this talk. We showed you that the anti-GLAST AXA1 and the AXA2 antibodies are novel tools for the convenient characterization of astrocytes by immunohistochemistry and immunocytochemistry, and they also can be used for flow cytometry. We can separate the astrocytes from dissociated whole brain tissue and distinct adult brain regions using the anti-GLAST AXA1 and the anti-AXA2 microbead kit. I also showed you that adult neural stem cells can be isolated from the mouse SBZ with our adult neural stem cell sorting and analysis kit in combination with the flow sorter MACS-Quant Tyto, and which leads to a high purity and a high viability of neural stem cells. And I also introduced you into the AstroMACS Medium that allows for cultivation of neonatal and adult astrocytes. Okay, I thank you for your attention.
Thank you, Melanie, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, our first question is, how many astrocytes can you acquire from one adult mouse brain, and what is the viability?
So from one adult mouse brain, we can acquire around 400,000 astrocytes, and the viability is usually 70 to 80%.
Great, thank you. Okay, your next question is, how long can you maintain astrocytes in culture with AstroMACS Medium?
We tested cultivation for 2 weeks, but I think it's not a problem to cultivate them longer. I think 3, 4, 5 weeks is not a problem, and maybe even longer.
Great, thank you. Okay, your next question is, does AXA2 antibody or GLAST antibody crosstalk with humans, and can they be used to stain or isolate human astrocytes?
So the anti-AXA2 antibody is not cross-reactive for human, but GLAST is cross-reactive. And the anti-GLAST antibody can be nicely used to stain astrocytes in human brain slices. But the isolation of human astrocytes is really tricky. So human brain is very, very difficult to dissociate, and our GLAST antibody is not compatible with the adult brain dissociation kit, as this kit includes papain, and the GLAST epitope is sensitive for papain, and therefore we have to use a trypsin-based dissociation, and so we do not recommend to use a trypsin-based dissociation for dissociation of human brain, as this is really, really difficult to dissociate. So therefore, we cannot recommend to use the anti-GLAST antibody for isolation of human astrocytes. But that's not a question of the antibody, it's more a question of the dissociation, which is really, really difficult in case of human brain.
Thank you. Can you isolate astrocyte from the hippocampus or cerebellum from one adult mouse? And if yes, how many cells can you acquire?
Yes, that's possible. So we have a special protocol for the dissociation of such small brain regions. And then it's also possible to separate the astrocytes by magnetic cell sorting. And from one cerebellum, we can isolate around 100,000 living astrocytes. The same is true for the isolation of astrocytes from 2 hippocampi of an adult mouse brain. We also obtain around 100,000 living astrocytes from the hippocampi of one adult mouse brain. So, and the viability rates, they are between 70 and 90% usually.
Thank you. Can AXA2 antibody and GLAST stain astrocytes from old mouse brain?
Excuse me, can you repeat? If they stain astrocytes in very old mouse brain, was the question? Yeah. Yes, they can. Yeah, they can be used for staining of also very old mouse brain in case of brain sections. So the expression of AXA2 and GLAST is not downregulated. At least we did not see a downregulation. And AXA2 can also be used for separation of astrocytes from old mouse brain in combination with the adult brain dissociation kit for dissociation.
Great, thank you. Okay, your next question is, when will you choose to use AXA2 antibody, and when will you choose GLAST antibody for astrocyte staining?
So that really depends. So both were shown to express the majority of astrocytes in principle. So we checked co-expression of both markers with other common astrocyte-specific antibody, and we saw that they all, in all cases, detected the astrocytes that were positive for other markers like GFAP, S100 beta, glutamine synthetase, and so on. So both are suited to detect astrocytes. So I would, the advantage of the GLAST antibody is that you can combine it with intracellular markers better than AXA2, because AXA2 is sensitive for detergents. It cannot be nicely stained anymore when the cells were treated with detergents, and then the GLAST antibody is preferred, as the GLAST staining is not affected by detergents. But both can be used for neonatal and adult mouse brain for staining of astrocytes, and in case of, if you want to detect astrocytes in rat and human brain, you have to use the anti-GLAST antibody, as the anti-AXA2 antibody is not cross-reactive.
Great, thank you. Can GLAST and AXA2 antibody also be used for Western blot analysis?
The anti-GLAST antibody also works for Western blot. In case of the anti-AXA2 antibody, we saw that it's difficult to use for Western blot analysis. So in that case, we would definitely recommend to use the anti-GLAST antibody.
Okay, your next question is, can you also stain or isolate adult neural stem cells from other brain regions?
So, for example, the dentate gyrus. So the dentate gyrus neural stem cells, they need another marker combination. So this marker combination that's here included in the kit that I showed to you is really optimized for the analysis and separation of the neural stem cells from the subventricular zone. So for the dentate gyrus, there's another marker combination needed. So therefore, we do not recommend to use this kit for the analysis and isolation of neural stem cells from the dentate gyrus.
Thank you. It looks like we have time for one more question. How many neural stem cells can you acquire from the subventricular zone from one adult mouse, and what is the viability?
So as I showed to you, we can isolate around 50,000 neural stem cells from the SBZ of 5 mice, so that means 10,000 neural stem cells from the SBZ of 1 mouse. That also works, so we showed that you can use the SBZ of only 1 mouse brain and then do the Tyto sorting. And the viability is again more than 90%.
Thank you again, Melanie, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.